Hello, my name is Dillon Heffernan, and I'm a high school researcher in the Duke University K-Lab. The K-Lab primarily focuses on mouse retina development. So a little bit of background before we get started on the retina. The retina is the phototransducing component of the eye, meaning that this is what turns light images into electrical signals that will later be sent to the brain by the optic nerve and will be processed into visual images we see every day. My project primarily focuses on two specific cell types, the astrocytes and the microglia. The astrocytes have been shown to be very important in providing a template for the blood vessel growth or angiogenesis. And the second cell type are the microglia, and these have been shown to be very important in regulating the number of astrocytes. So astrocytes actually overproliferate by P5 and are colored by over threefold by P14, and we think that microglia play a factor in this. So it's been shown that any disruption to the number of astrocytes that are outside of the optimal range leads to a disruption in the template and later leads to retinal damage. A common example of this would be a baby introduced to the NICU, which is a high oxygen environment. The astrocytes can actually detect the variation in oxygen, and this will disrupt the template. So we wanted to further investigate this because little is known about the actual molecular and genetic processes of this. And through this, we wanted to do RNA sequencing to target specific genes, and through this, we discovered ApoC1, a novel gene, and we wanted to further investigate this. So our hypothesis was that phagocytic microglia express ApoC1 during the astrocyte death period, P5, but not afterwards, which would be P17. So we compared two different experimental time periods, P5 mouse retinas and P17 mouse retinas, and wanted to analyze and quantify the ApoC1 gene expression. So onto our results through in situ, we can see that C1QA is green, and this is just a pan-microglia marker, meaning that it marks all the microglia in the retina, the nerve fiber layer. ApoC1 is in red, and these are the cells. This is the gene that we're looking at. These cells are expressing ApoC1. And SPP1. So SPP1 is a more studied common known gene expressed by microglia that specifically phagocyte astrocytes. This is why we wanted to compare ApoC1, our novel gene, to SPP1. Emerge signal just shows all emerges. So we can see for P5, we can see overlap between ApoC1 in the central, middle, and peripheral regions. And in P17, we see absolutely no overlap, and even more, we see no ApoC1 gene expression or SPP1 gene expression. And this helps lead us to our conclusions that there are more double positive SPP1 and ApoC1 gene expression in P5, and there are no double positive in P17. And our second conclusion is that ApoC1 expression in microglia appears to increase in a central to peripheral gradient, meaning that as we work towards the peripheral of the retina, there is more ApoC1 gene expression. And this is what we would expect to see because at P5, the wavefront, or where the tip of the retinal angiogenesis is where we expect to see the most astrocytes, and this is near the mid-peripheral. So more astrocytes signals more engulfment and more phagocytosis, which would mean more ApoC1 gene expression. At P17, angiogenesis is 100% completed. There are no astrocytes that reside near the wavefront, meaning that there would be no engulfment. So we hope that this later in the future could help address some of this retinal damage problems that occur in the NICU and help address some of these issues. So yeah, thank you.